To prepare the DNS reagent, 0.5 grams of 3,5-dinitrosalicylic acid are added to a 50 ml volumetric flask. In the lab, more substances like potassium sodium tartrate, phenol, and sodium sulfite are added to increase the stability and sensitivity of the reagent. To this, 10 ml of a 2 molar sodium hydroxide solution are added. The yellow sodium salt of the dinitrosalicylic acid is formed immediately. The beaker was washed out 2 times with some distilled water to add the rest of the acid. The solid dissolved very slowly, so the flask was filled up further. In the beginning, a wash bottle can be used for this, but at the end a pipette should be preferred. It is filled up until the lower meniscus of the solution hits the mark. Then the flask is panned occasionally until all of the solid has dissolved completely. This took about 1.5 hours. Next, a dilution series with glucose was prepared, where the concentration was decreased to half of the amount in every new sample, beginning at 1%. In the quantitative detection, the samples are cooled with ice after the heating to prevent further reaction. It can be seen that the intensity decreases with a lower concentration. What seems to be wrong is that the concentration at 1% seems to be lower than at 0.5%. A possible explanation might be that the concentration of glucose was too high for the detection. Usually the concentrations are kept between 0.1 and 0.01% for the measurement. In the lab, the samples are then diluted and the absorption is measured at a wavelength of 540 nanometers.
Hi, today we'll be learning how to use the UV visible spectrophotometer. Before we begin, let us discuss the different types of cuvettes that you could use. These are the standard plastic cuvettes, which will be used for normal lab work. These, however, are quartz cuvettes. They are very expensive and should only be used for highly corrosive substances. Before turning on the machine, ensure that there are no cuvettes in the sample compartment. Turn on the main switch and the power switch, located at the back of each unit. Wait for the machine to run the startup sequence and do not open the compartments during this time. To prepare the cuvettes, wash the cuvette with deionized water and rinse it with the solution you are about to use for the analysis. The cuvettes have 2 translucent sides and 2 clear sides. When handling the cuvettes, always handle them by the translucent sides and keep the clear sides clean at all times. Use a tissue paper to thoroughly wipe the clear sides of the cuvettes before use. Now, we'll learn how to make a calibration curve. To do so, you'll first need to prepare a micropipette, standard flasks, and cuvettes containing solutions of different known concentrations. Insert a blank cuvette into the machine. Select Quantitation, Sample Compartment, and adjust the drive cell number to the number of cuvettes you are going to use. Return to the previous page, select Measure, enter 1 lambda, and adjust the required input wavelength. Now select Method, Multi-point, and adjust the number of standard solutions. Press Enter and select Zero Intercept as off. Select Auto Zero to begin the calibration process. When the process is completed, replace the blank cuvette with the prepared cuvettes of standard solutions. Press Start and input the concentration of the solutions for the corresponding cuvettes. Select Multi-Cell Sequential Measurement and press Start. When the analysis is complete, select Calibration Curve to view the graph and Equation to view the equation of the graph. The UV visible spectrophotometer may also be used to find the absorbance of an unknown sample. To calibrate the machine, place a blank cuvette in the machine, select Go to Wavelength, and select the desired wavelength. Then select Auto Zero to calibrate the machine. Replace the blank cuvette with a cuvette containing the unknown sample, and the absorbance value will be shown on the top right of the screen. To analyze the absorbance of multiple unknown samples, select Photometric, Photometric again, and Sample Compartment. Adjust the drive cell number to the number of unknown samples you intend to analyze and return to the previous screen. Calibrate the machine with a blank cuvette as previously done. Insert the multiple unknown samples and press Start to obtain the absorbance values.